Okay, so the slide set, I thought that maybe the most relevant slides are the first ones about the allometric scaling laws and stuff like that, just because that's a common question, and it's also one of those big picture kind of things that you can understand without knowing a lot of background details. And then things about the brain and how it functions as a machine, sort of, how many watts it is. And of that wattage, what percentage cognition uses. So you'll find, like in slide 62 and the ones beyond, the brain itself takes 25 watts uses 25 watts, but only 5 watts of that is actually used for cognitive processes. So it's a remarkably efficient entity. So like the slide behind me, which I think is slide 62, compares the brain with the blue gene computer. And the blue gene computer is, uses thousands of times more energy than the brain. And yet is not quite as fast. The blue gene computer is at 10 to the 15 operations per second, whereas the brain is at 10 to the 60. So I thought those were like areas of the slide set that had memorable concepts, but were within your capacity to understand. So it's just kind of cool thinking of the size of an animal or plant or whatever, and how much energy it takes, and so on, and what those ratios are. So those are in the early slides in the slide set. Let me just show you what I mean by that. So this whole idea about energy consumption in organisms, and the simple idea that respiration is a form of combustion is a little bit like burning something in a bonfire, only much better organized, I guess. And then this idea of the allometric scaling laws and what they're like in plants and animals and single cells. Yeah, and then there's a lot about microtubules here. Microtubules are one of Jack's very strong interests, and so this slide set gets deeply into them and so on, and mitochondria and energy production and photosynthesis. Yeah, so then if I go ahead, see if I can find. Around slide 60, we get to the idea of quantum things explaining brain function. Yeah, so it starts here. And I didn't include all of the consciousness slides because that's probably the most speculative part of this. So you just have sort of the earlier parts of that. What makes the brain special? What are the various ways of explaining information processing in the brain? How can it work with so low power compared to computers? Now you'll recognize this slide is very similar to what David Pierce said about the binding problem. In other words, both Robert Epstein writing here and David Pierce believe that you cannot really compare the brain to a computer sort of at all, that you're missing something big if you think you can. Of course, comparing a brain to a computer is by far the simplest way for most people to think of what the brain does, right? So when Robert Epstein and David Pierce tells you not to do that, you kind of secretly thinking, well, I don't have an easy alternative to comparing the brain to a computer. So I think I'll still do it. So that's probably what you're still thinking. But it is an interesting idea that we may be way off doing that. So, Simon, did you have thoughts about the approach to this second half of the slide set?
So I see you like trimmed it down a little bit.
What?
Have you trimmed it down? Can you hear me?
No. So it's only trimmed down in the sense that it started at a certain point, but then I thought I was going to leave out the slides that just didn't work on this template. But there aren't any slides that don't work. So I was able, just by changing the colors, I was able to make all the slides work. Yeah. So I didn't really, I didn't have a rationale for removing any slides. So I didn't. So yeah, so that's why this second slide set is so huge. And that doesn't mean that the students can't remove slides. They obviously can, but I just sort of ran out of time. I kept thinking, well, the slides where we can't see any text, I will leave those out. But then I just had to change the text to black and change the font size, and then I was able to get them to all work. So
I mean, for the second section, I think there's like 5 main topics that Jack Kaczynski focuses on. So the first one was like the quantum brain, which is like this slide right now. So his first point was like, what makes the brain special? And then he had a bunch of questions in the previous slide.
right. Yeah.
And then he has a lot of like theoretical calculations here, right? Talking about like the number of neurons versus the number of synapses. and then just the sheer efficiency of the brain, that it only needs like 25 watts of power compared to 1,500,000 of a supercomputer. Now, still under the quantum brain, there's two more topics he talks about, three more, sorry. One is consciousness, then memory, and then he answers the question, is our brain a quantum computer? This is still the first topic, which is the quantum brain. I'm just doing a quick overview of like the second section.
Yeah.
And the second topic he talks about is brain diseases. And so the main disease he focuses on is Alzheimer's disease. And he links Alzheimer's disease to microtubules, which is his like favorite structure in the cell. And so he relates microtubules to quantum biology. So that's the second topic, brain diseases. And the third topic he talks about is quantum anesthetics, which is a really, really short topic, but he talks about, again, how microtubules could contribute to anesthetics. And then the fourth topic he talks about is quantum psychology. So how psychology could be related to quantum biology. And then the fifth topic he talks about is quantum embryology. So like, is there quantum effects in the way we grow from one egg to the shape we are now, like one egg to 4 eggs, to 8 eggs to 16 eggs, or not eggs, sorry, like 16 cleavages.
Yeah.
Right. So yeah. And then as a bonus, at the very end, he talks about like bioelectrical medicine. Like if we could possibly one day gather information straight from the brain through an interface as like a bonus topic. So yeah, there's like 5 main topics to the second section and then 1 bonus topic.
So Eduardo is not able to be here today. So
what I could do is like send everyone this Word document I have that like summarizes the first and second lectures of Jack Dizensi. Do you think that would help or?
Sure. Yeah.
Okay.
I think so.
Okay, well, I'll just send it to you, Dr. Solez, and then you can like.
Yeah. I'll send it
out
to the students. Yeah.
I had a quick question.
Yeah.
I guess I'll take Eduardo's place where he usually asks all the questions, but this is kind of going back to I think, I don't know what day it is anymore, but whatever the last class was, we talked a little bit about olfactory senses, and I was wondering, and like the kind of quantum side of that, I was wondering if you could speak to that a little bit more. And also something that I was wondering about when I listened to that lecture was how when we smell certain smells, we associate it with certain memories, or even like when this is gonna sound kind of weird, but like certain people's homes that I've been to when I was a kid, there are certain smells that I associate with them. And like when I smell it, it triggers a memory, or I associate it automatically. So I was just wondering if there's anything quantum about that, or I know there might be like other scientific research out there that kind of explains.
Well, see, this is sort of the area that Simon originally was interested in, and when he was a student in the course, that's what his presentation was on. And it substantially added to what we were teaching in this area of the course. So yeah, it had quite a big impact. So that's a good question to ask Simon about the relationship between smells and memories and quantum.
Is that specifically your question? Like how our olfaction is related to our memory? Is that your question?
Yeah, and even kind of shedding light as to like the quantum side of things, if there is a relationship to that. If not, I'd just be interested in learning about the relationship between olfaction and memory as well.
I see. I don't think I really went over that topic. I didn't research that topic that in depth. Although learning about that topic, I realized that our memories, I think Dr. Solez had a slide, like you're not going to be able to find Beethoven's 5th symphony or whatever stored in our brain. as I was researching, I began to realize like our memory is like it's contained in all our senses, right? Like whether it was your last memory eating dinner or meeting your friends, it wasn't just the visual of seeing your friends. It was also the memory of hearing their voice. The location was it cold or hot, what odors you smelled. So like our memory is like all these senses, right? And all these senses, you know, like our brain is organized with all the different centers, right? So in fact, like our memories are like coagulated in all places of the brain at once. You know what I'm saying? So it's not like one single location. And I also know that odor in particular is like strongly related to our memories. It's like strongly linked. I remember in grade 11, the chemistry teacher was like, maybe you guys should like bring different fruits as you memorize different facts. So when you smell the fruit, you memorize. I don't know if she was like talking out like to be funny, but like she suggested that. I don't think she was kidding. So I thought she was kidding, but now I realize that there was some truth to what she said. Like smell is one thing in particular that really links to our memory. But whether there's a quantum side to that, I think there most definitely is a quantum side because Jack Kaczynski's constantly saying all our memory is like quantumly related. So I think without a doubt, there would be some quantum connection, although I don't know the specifics.
See, the things that we're able to do with olfaction and sight and memory, I would think, are just not explainable through classical physics. You know, like we're able to see a single photon. Well, that doesn't make any sense that we would be able to detect a single photon. And it can only be explained if sight is quantum. And similarly, like maybe not our nose, but like the nose of a dog, or you know, there are other species that have much better noses than we do, and what they're able to do with that is just amazing. And in terms of, you know, specific identification. Yeah, and Taryn says, I took a neural anatomy class a few years ago. The way sense are processed is different from other senses. The way they explain it is that the olfactory bulb is directly connected to the hippocampus where memories are stored, and the amygdala where, you know, emotions are. So it's like a direct connection between olfaction and memory and emotions. That's just how the brain is wired. That's what Taryn's chat comment says.
I think that's a great point. Yeah, very good point.
Yeah.
Along the same line, I can't remember if it was he talked about it or not, but the fact that like we will meet someone, take in like all of these things that we're not actually like consciously thinking about. Like we can pick up different like body language and all these other things and put it together and like get like, I don't know, like a vibe or like a like
yeah, like creepy, non-creepy, you know, like sort of immediate senses. It's just like something I'm aware of. You decide when you begin watching a video whether or not you're going to keep watching it within a few seconds. I mean, it's not based on just one thing. It's a whole bunch of things. And it's the same when you meet people. You're immediately deciding whether you like them or not, whether they represent a risk to you, you know, whether you should start looking for an exit in a flight path having met this person, or, you know, plan future things with them. So all those things in a few seconds, you're putting that together. And I think without quantum, you can't really explain how that could work.
I would like to add something I thought about actually for the quantum biology. What came to my mind like is about the nanotechnology, if that actually is representing the quantum biology. Do you think, for myself, I don't have I don't have a real big background about nanotechnology, but I mean, this what came to my mind when we talk about the quantum biology. Do you think the nanotechnology is part of it or representing it, or it's actually something far away?
Well, it is part of it, but nanotech, I'm sorry that in this term, we're not really getting any nanotech teaching directly. It is something different. There are special phenomena that occur and when you get down to very small scale, and so there's sort of, just like there are different rules for quantum versus classical physics, so there are different rules for the way molecules interact at very small scale compared to bigger. So yeah, the two are sort of related, but they're different also.
Does the nanotechnology actually use the rules of quantum biology, or?
It does, but it doesn't have to, and it didn't begin that way. It began with that quote that I gave you before. There's plenty of room at the bottom, that, you know, is a whole world that is there to explore what's going on at this sub-microscopic level of things and what the rules are there. So you can look back at the nanotech lectures. You know that some of the most popular lectures in the course, we just look at the YouTube channel. There's a line there that is the most popular, and I think there are nanotech lectures there that are like the third or fourth most popular ever in the course. So you can easily get farther into nanotech. A lot of the things that we ended up showing are things that require near absolute zero temperatures and, you know, sort of elaborate setup to make those things happen. Even simple things like using molecules to spell IBM, you know, it doesn't seem that complicated. So any of us could do it. You just put a molecule here. It turns out that it's not quite that easy at that scale. So it takes, you know, hours and hours to set up these things that look simple in the end. So like the things that were always sort of intriguing to me in nanotech is how real is this? Because even the people teaching it use science fiction images for their first slide. Like they say, well, now I'm going to tell you about nanotech. And they show you this nano louse, or whatever you want to call it, a sort of fictional thing that doesn't actually exist, going around in, you know, the bloodstream, fixing other cells, or, you know, seeking out cells you don't want and killing them and stuff like that. And this is something that we certainly believe will exist in the future, but nano louses like that don't really exist, but they're really cool to look at. You think, wow, it's an amazing machine and so tiny, you know? And so there's no other field of science that I know of where the practitioners of that field begin with something fictional on the very first slide, right? But that's what nanotech is like, and everybody seems to do that. because the reality of what we're able to do with nanotech now just isn't as visually interesting as these theoretical nanotech things for the future. And then there's something else that probably is not worth your time at the moment, but cryonics, the belief that you can freeze people and bring them back. And you know, there are people now being frozen. But what is the theoretical solution? It is the idea that these nanobots will go in and fix everything. Like what I can tell you about the reason that cryonics now doesn't work is that when you freeze an entire human body, you get these big cleavage planes, like big rips and everything, rips in muscles, rips in skin. So if you did succeed in bringing the person back to life, be worse than being dead, right? Because you've got all these painful defects in skin and muscle all over your body. And, but the people who support this say that the nanotech, the nanobots are going to go in and fix all these things, but that's impossible because these are not at the microscopic scale. They're big sort of cleavage planes in skin and muscle that you can see. They're just gaps, you know, they're macroscopic gaps that would be clear to anybody from the ice crystal formation. Even with very fancy ways of freezing, we can't freeze anything larger than about a rabbit kidney and have it come back. So that larger organs like human organs and certainly an entire human being, you can't actually bring them back. But when you talk to the believers about that, you say, well, my uncle is frozen away and nanotechnology is ready, we're gonna bring him back. And it's hard to tell those people that no, that's not true. There's nothing that nanotechnology could do to bring this relative back because the problems are not microscopic. The problems are like macroscopic. And yeah. And then there's some people where it's not their whole body frozen, but just their head. But even that is a problem because, you know, you're getting serious damage to all the structures in the head during the freezing process. So the bottom line is, I don't want you guys to learn too much about cryonics because it's really something that's like a fraudulent area of science, right? You have people paying money to keep their relatives frozen for decades, where it's not going to be possible to bring those people back. But I do want you to understand that there is this fictional aspect of nanotech. It's not just this use of things that don't really exist for the first slide, but also the idea, this strange relationship between nanotech and cryonics, where nanotech is going to be the solution to bringing all these frozen people back in a pain-free, functional way. And that's just not possible.
I had a question about something that you and I, Dr. Solez, talked about at the very beginning, which is music and the brain. And I was wondering if there is anything kind of going on at a quantum level that we've discovered in those areas.
Well, I would imagine so, but it's not something that I have looked into in great detail. Simon, do you know anything about quantum and music?
Definitely not. But I'm all ears for sure.
Yeah. We both believe that kind of connection will be found, and yeah, but we don't have any evidence. Yeah.
Ooh, although I'm curious, like I remember my high school teacher asked the whole class this, so I can ask you guys all this. Like, would you guys rather lose your eyesight or your hearing? Both pretty bad.
Well, to me, that seems easy to answer, because many deaf people believe that, the world of the deaf is a separate culture and an entirely satisfactory one, that sign language works and that they are able to, convey information just as well as non-deaf people. And although those of us who have hearing probably don't feel like that, but certainly there aren't any blind people who would tell you that same thing. That yeah, the culture of the blind is just a separate culture. And if you understand it, well, you understand that we don't need sight. There aren't any blind people saying we don't need sight, whereas there are deaf people who say we don't need sound. I haven't had a sound my whole life, and it's been fine. And sign language is getting better and better, and yeah, it's just yeah.
I remember back in that moment, I was like, eyesight, of course. Like, it'd be impossible to live without eyesight. But I think like now I realize like there's a lot of emotion when it comes to like sound compared to eyesight. Like hearing your parents talk, hearing your friends talk, versus seeing your friends. Like I think like hearing and speaking is a huge interaction point compared to just like watching. So that's what I think now. So I think it's really hard. But I don't know, how does this relate back to your question, right, about music? I think my point is like music is like a hugely emotional like aspect of our brain. So yeah.
I mean, I can talk in that there's a chance that I almost lost my eyesight at one point. And honestly, that's nowhere as bad as if I felt like I was losing my hearing, because that's like almost all of my free time is just spent listening to music, and that's how I get like a lot of like having feelings or like feeling connected to the world sometimes, where like I had to live with like one eye, like an eye patch for a couple months, and like it sucked, but I'm also really blind. Like if I take these glasses off, I have horrible eyesight, like almost enough that like my optometrist is like, you're running out of options. But I like, I don't know, it's something that I feel like I could live with a lot more, because like you said, there's less like, for at least me, there's less emotion attached to it, where like, I guess, because I think like a song that's beautiful is a lot more interesting than like a picture. But I think that's also different people's, I think it's like a very generalized, like a specific thing to each person.
Yeah.
So I don't know if you can generalize which each person, what would the best option be for everyone.
So, in the future and all that jazz, we're putting three things together. We're putting science and poetry and music together. And you say, well, what do you mean together? Aren't you doing them sequentially, like you do something about science? Well, no, we're really discovering that it works a lot better to do them together. Like I think you guys already have the link for the future and all that jazz video shoot that we did on the 6th. And so the first part of that is me introducing what the future and all jazz is, and the fact that we're going to do an album in 2021. So if you just listen to me pitching it, and then Mallory Chipman and her band start with this song about an artificial intelligence entity talking about she needs to be treated with, you know, respect, and she's worthy of as much regard as a human being and stuff. It works much better to actually merge those two. That, like, the start of that song, before the words start, it doesn't really grab your, you know, attention totally until they actually start with the words. So what we're doing is mixing my intro with the first part of the song, so that we're sort of combining the two. Makes my intro more interesting. That suddenly there's this unexplained sound, and then you realize, I guess it's the start of a musical piece, and you know, what is that? And then, yeah, at a certain point, we go from the audio of that in the background for what I'm saying, and then change over to the full video, and at exactly the moment where Mallory Chipman sings the first word, so you can kind of figure out what's going on. Similarly, we have poetry about the modular synth, and so you know that the modular synthesizer, it can really replace human activity exactly, just like you've probably heard me talk about this before, but get, you know, musicians performing music, right? And the modular synthesizer then can kind of store that, and then the musicians stop, and the modular synthesizer puts those bits of music together in new ways. Sometimes it's much more beautiful than the way that they were before. And there's no human being doing anything at that point. It's completely computer based. Anyway, these are some of the ideas that we're playing with in the future and all jazz. The live stream we'll have done in November sometime, and it'll be part of the album later, which will have 7 other sections. So the live stream will be approximately 1/8 of the overall album. Yeah, anyway, that's, and you can think about the potential audience in 2 ways, right? So it's a pretty unusual thing we're doing, right? So you can say, well, maybe the audience is 0 because nobody out there looking for this. You're providing something that nobody's looking for. On the other hand, what we're providing is a sort of mixture of things that have a large audience, right? It's a large audience for music, for science, and for poetry. And so properly done, this combination of those 3 should also have a large audience if we do it properly. Anyway, that's our story, and we're sticking to it. Yeah, I mean, it's sort of like, you remember the part at the end of one of Dr. Tuszynski's lectures. He was saying all the textbooks will be rewritten, you know, to be quantum, and that was the part that the IBM people liked and urged all their employees to watch. And so that's why we have, like, you know, whatever it is, 6,000 hits, 6,000 views there.
So
I have a question about something else.
Yes.
If you wanted, okay.
Sure.
So there's a lot of things that we're not really sure, like, because they're trying to find the link between like biology and quantum in this. If say like, would it give a lot of evidence to biological things being quantum if they could only be replicated by a quantum computer? Like if we took a normal computer and tried to figure out, like, run a same type of kind of thing similar to what a body would do, but it can't be done on a normal computer, but you can do it on quantum computers, could you then kind of say that?
I think that those are kind of apples and oranges, but with the same words, right? That it's true that quantum computers do different things from classical computers, and quantum physics is different from classical physics. But whether you can replicate some human activity on a quantum computer versus a classical computer does not really represent proof that it is actually quantum. I don't know. It's maybe suggestive, but it isn't proof. Because whether a computer can run something doesn't mean that in the human body that's how it works, the same as what the computer's doing. So if a quantum computer can simulate some human activity, that doesn't mean it's doing it the same way as we do it. Simon, what do you think about that question?
I don't have much comment to it. I was thinking of another question.
Okay. Yeah.
But before that, like, did you have any thoughts on that, Dr. Solez? Like, did you have more to add, or?
No. I just wondered whether maybe you held the exact opposite opinion. But, yeah.
I think I'm neutral on that.
Okay.
Yeah. My question was on your cryo freezing thing. Like, is it just because the water expands when frozen? So, like, if there was another solvent that humans could be placed, and I know you're like a pathologist, you know, like the cell. Like,
yeah. So there are many ways of freezing things where you end up with a sort of glass-like ice rather than just old-fashioned regular ice. Regular ice, you know, has these big crystals that sort of break cells apart and stuff. But even with those cryoprotectants, we still don't have the science to perfectly freeze things, big things bigger than a rabbit kidney. And the story of the rabbit kidney is sort of interesting. You say, well, Dr. Solez said that you could always freeze a rabbit kidney perfectly. No, that isn't what I'm saying. So 7 out of 10 times when you freeze a rabbit kidney, it doesn't work at all. It just doesn't work. But 3 times out of 10, it does. And we don't know why. You can't reproducibly do it. So that's like the limit of being able to freeze something and then bring it back and have it function. But we don't really know what parameters account for the fact for the 3 out of 10 times when you can actually freeze a rabbit kidney and it'll come back and work. So probably someday we will know that, but we don't know it today.
So like, is there such thing as like having the solvent be like oil-based or something, so it doesn't? Is that a thing, or completely not a thing?
Well, I think what you're talking about there is probably super cooling, right? Cooling, but not freezing something. But that isn't what they're trying to do. You see, they're trying to actually freeze something, because if you just super cool something, it isn't stable over many years and that sort of thing. So they're trying to find a permanent way to keep these human bodies so that when you find the cure for whatever ails the person, then you bring them back to life, cure their disease, and they're all good to go. You know, that's sort of a primitive way of thinking about it, but it really doesn't work. So that's fine.
And then my last point is, you talked about the 3 out of 10 chance, 30% chance at working. Like, if those companies are freezing 1,000 people, what if just one case works, right? It's like, statistically speaking.
Yeah, so I guess that's what they're saying. You know, how do you know that my uncle whose head is frozen isn't coming back? Just because the other 999 people of the 1,000 whose heads have been frozen are never coming back doesn't prove that my uncle can't come back. Maybe he'll come back and be completely functional, and boy, won't you be surprised, you know? So yeah, anyway.
Wow, parents talking about this skim milk, I've never heard of that. I think we need to bathe ourselves in skim milk. Maybe the crowd companies consider
that. Wow.
We're all just gonna start taking skim milk baths.
Yeah, when you're nearing, you know, the palliative care stage, then start chugging the skim milk.
Yeah, that's
Get it in the system.
Yeah, wow.
That's actually make me think about the cell culture work that we do in the lab. I was thinking, even if this process of freezing complete by the human body, when that comes to life, would that be the same? I mean, will that person still carry the same memories, the same, you know, behavior?
Yeah, well, you can do it in worms now. You probably know this elegans worm. So you can freeze them. And so you can teach them things and freeze them and bring them back, and they still know the stuff that they learned before. So the memory is maintained in the frozen state. Okay. So, yeah, but they're very simple animals. You know, they have a countable number of genes and molecules and stuff. And the world of the C. elegans worm is very simple compared to human beings. So that doesn't mean you could do that with us, but you can do it with them. Yeah. Because
I like when we do cell culture work, actually the cell lines that are actually can be generated up to like 60, 70 times in the cell culture. And this is just a one cell kind or a one cell type. They couldn't go that far without being transformed. Like they are transferred with a virus, or, you know, their genes are modified somehow to be stable. But when we are using primary cells that are actually from donors, we cannot grow that more than 5 generations. Then it would be over. Like it cannot go further. So, I mean, how is that related to freezing a human body? Like, would that be able to generate afterwards? Like, if there is a wonder something happened, would they be able to regenerate? Or, I mean, it's a big project, or a big, you know, question to think about. Each cell type in body human, how would that be functioning after freezing?
Yeah, well, I mean, that gets into bringing back animals that you may feel great affection for, but that don't exist anymore, like bringing, you know, Brontosaurus back, or, you know, T-Rex or something, right? Whatever your favorite animal from the past is that you really like to meet, you know? So if we had frozen tissue from like let's say that you really want to meet a mammoth, right? So there are areas of the world where there are frozen mammoths, and they probably weren't frozen in an ideal way. I mean, they probably suffered and were frozen slowly, right? It isn't they were quick frozen. These are slow frozen mammoths. But maybe, so you couldn't bring them back to life, but maybe their genetic material may be sufficiently intact that you could take a nucleus and insert it into another living cell and, get recreate a living mammoth through that, rather than bringing the original one back. But yeah, I mean, through genetic material, probably eventually we will learn how to do that. You might imagine that the first time that it's done could be something illegal that the person goes to jail for, right? And also clunky, where they don't really know what they're doing. So maybe the first prehistoric animal brought back will be an animal that nobody wants and creates enormous destruction, T-Rex going through cities and killing everybody and stuff like that, all because of somebody's stupid project that actually sort of kind of worked, but not the way they planned. Like the CRISPR-Cas9 thing in China, even when you look at whether the person is, immune to HIV, I don't think they are absolutely protected against HIV. So even that part of that study didn't really work.
What's your opinion on like ethics and like trailblazers? Ethics keeps everyone safe, but perhaps being so safe limits the speed of like advancements. But then too much advancement is unethical because you run the risk of doing harm as well.
Yeah, well, you can look at some examples. In vitro fertilization, when it was begun, was a very controversial thing, and there were all sorts of conferences about it, and people very fearful that this is a road we should not go down, and now it's completely routine. So I mean, it isn't always the same. There are some blazes, some trails that should not be blazed, right? We don't want to go down that path. No thinking, caring person should want to. Whereas others have promise and probably will figure out a way to do those things. And there's the whole thing about diversity. It's not just a scientific question, it's a kind of political one that goes up and down about how accepting of, diversity the population is and how they are defining it and what's acceptable and so on. But we imagine that eventually there will be sentient machines that are included, having similar rights to humans. So I think the question that you posed doesn't have just one answer. It depends upon the circumstance. So, yeah, Taryn says replication is also limited by telomere length. So our ability to replicate a cell with something dead is limited by how old or healthy that organism was before it died. Yeah, what Ashley says is also true. We don't know all the effects of in vitro fertilization. So on Thursday, Ishita Mogi is presenting to you, and I think you'll find it quite interesting. She's still figuring out exactly what she's going to teach you. But so that'll be at 2, the usual time. I didn't want any of you to think that we're changed the time of the course. We haven't changed the time of the course. It's just, this isn't the lecture proper. This is the discussion of the lecture, right? So that's why we're doing it at 3 rather than at 2. Yeah, and then Dr. Van Manen is doing 2 general lectures on ethics of the future. And then we're having bioartificial kidney lecture. Many of you will be aware of Shuvo Roy's lecture on the bioartificial kidney from 2015. That's like the second most popular video in the course. So his physician collaborator it is going to tell you what's happened from 2015 to now with the bioartificial kidney. And then you're getting a bit of anthrax and bioterrorism, and then there's reading week and the midterm exam. And then finally, oh yeah, the other thing I wanted to mention, thank you for getting back to Rebecca Hu about the photography for the cardboard cutouts. The other scheduling thing we need to figure out is that last lecture in the course, just before we start the student presentations. That lecturer lives in India, and if we do this at the usual time, it's like 2 or 3 a.m. his time, which is not practical. So I need you guys to let me know, like, if we did it at 9 a.m., how many of you have a conflict at that time? I don't think we could get all of you, but I'm just wondering, if we can't get any of you here, just me and him, then it won't be very satisfying. So anyway, I'll write to you in more detail about that. He's like the world's expert in analyzing networks, like scientific networks. And he has analyzed the network of transplantation or transplantation pathology, the field that I sort of lead. And so he's going to talk about that, the analysis of scientific networks and what the advantages of that are. He has probably the best computer program for doing that. So I think that'll be an exciting last teaching session. And then you have to decide about yourselves and how you want to divide things up. In other words, over how many days do you want to do the student presentations? And which of you want to go earlier and late, or do you want, yeah, I'm hoping that all 8 of you don't want to do it on the same day, in which case I will be extremely fatigued by the time that we have presentation from the last student. So that's the disadvantage of you know, having most of them on the same day. So those are other things for you to think about, scheduling wise. So any questions? Okay, yeah. Thank you, Caitlin. All righty, so. Anything else? Anything else you want to talk about? Thanks to Simon for helping us through this part of the course. And I know it has, what?
I just meant I send you the doc so you can send it to everyone. It might help. I hope it helps.
Cool. Yeah. You know, there are some things about this area that even I don't understand. I don't understand why Jack's PowerPoint worked for me when I received it and doesn't work now. I mean, there's probably some obvious way to explain how a PowerPoint master that worked in 2019 wouldn't work in 2020. I don't know, but anyway, I think Simon also found that somewhat discouraging, that we thought he had a really good slide set that looked just like Jack's lectures, and then suddenly we didn't, you know, we had the slides, but not the proper background. So I'm still a bit mystified at why that happened, but it did, so there. Okay, any other questions? So we'll see you on Thursday, and thanks for taking part in this session today then. Okay, bye-bye.
Thank you, bye.
You're welcome.